Okay, so we're going to begin today by picking up where we left off. We had talked about how lymph travels through the body here. Now we're going to start talking about some of the moving parts of the lymphatic system itself. To wit here, these will be things like lymphoid cells. Now these, of course, are primarily things like lymphocytes. Right? We've talked about lymphocytes before. We're going to talk about them more today. We'll spend a huge amount of time talking about them in the future. For right now, we're going to just kind of leave it at they are divided into two kinds. Okay, T cells, right, which we may remember as T lymphocytes from before, and B cells. And these guys are going to be involved in what we're going to call the active immune system. And the active immune system is the one that is involved in fighting specific pathogens. So when you get a vaccine, for example, or an inoculation, or you have a sort of an accidental vaccine, you get a cold or something, these are the cells that are going to be engineered to have a response specific to that. Okay, so one of the reasons that you have to get a flu shot every year, right, is that every different strain of the flu is slightly different, and the T cells and B cells that you had active for the last strain of the flu are not the same ones, and they are completely and utterly ineffective against these new strains, which is why things like animal transmitted diseases are so very scary, right? Because they are novel, they are brand new, and we have no immune response to them whatsoever. So you probably don't remember this, but last time we got hit with one of these things was H1N1 of the swine flu. We almost got hit with SARS. Thankfully, SARS is really bad at spreading, and so it didn't spread very far. But this new one, again, this is another novel disease that we have no immunity for because it was never in humans prior to this point. And so while we have a wonderful active immune system, they are totally useless at fighting off the infection the first time. Why they are so specific and why they are useless against new things is that what they are looking for are antigens. Okay, and an antigen, we talked about antigens before when we talked about the circulatory system. The idea here that every cell in your body has a specific name tag that marks itself off as being your cell, and more specific than that, your liver cell or blood cell or whatever else, that's an antigen. And so we know which things to get rid of by looking for the name tags that don't match, right? You can't just take your Burger King uniform, go to Popeyes, hop behind the counter, start working, right? You'll be thrown out. This is the same basic idea. So anything that doesn't have an antigen that says, hey, it belonged to you, this is where we're going to start having B cells and T cells active. So there'd be things like bacteria, virally infected cells, not viruses themselves, but virally infected cells, cancer cells, a bad blood transfusion, any of those sorts of things. They're going to respond in slightly different ways. T cells are a little more varied. So some kinds of T cells actively attack and destroy things. Some kinds of T cells are involved in activating other T cells, activating B cells. So they have kind of a multi-layered role there. Again, we'll talk about more when we talk about the immune system. B cells, these become plasma cells, which you might remember from before. The idea behind a plasma cell is that they make antibodies. And all an antibody is something that sticks onto a specific antigen so that we know to get rid of it, right? T cells don't actually attack antibody-labeled cells. That's for something else. So they are going to go out and manage and attack things based on antigens by themselves. B cells are for macrophages, monocytes, things like that. Other lymphoid cells here are going to be things like macrophages, right? Again, macrophage, big eaters, this is what it means in Greek. They go out and they find things that are foreign, right, that are marked off by an antibody, and they destroy it. They literally swallow it, digest it, crap it out the other side, and get rid of it. A similar idea to a macrophage is something we're going to call a dendritic cell. A dendritic cell is going to swallow most of a pathogen, but it's going to leave a little part of it hanging out. Okay, it's going to take the antigen from that pathogen, whatever it happens to be, and it's going to have it still kind of sticking out. And the reason they're going to have it sticking out is so that they can deliver it to our lymphoid cells, to our lymphocytes, so that they can recognize in a safe fashion what the foreign antigen is. So they can begin to work on it without having to be potentially infected themselves. Finally, here we've got some structural cells here. These are going to be the reticular cells. They make what's called a stroma, and we're going to use this term stroma quite a bit. It's basically just a net, right, or kind of like a hammock that's there to support other cells. So if we look at the inside of, in this case, a lymph node, we've got this big hammocky network of stroma with all of our cells within it, both lymphocytes here, these will be B cells, and also macrophages surrounding it. When we start putting this stuff together, we get what's called lymphoid tissue, okay? And basically the idea behind lymphoid tissue is that it is a place where these guys can come hang out, they can grow up, they can mature. It's very much like college or maybe like a boarding school for lymphocytes. And the reason being that because what they do is so potentially dangerous, right? They're telling what cells of our what cells in our body that need to be destroyed, or they're actually destroying them. The potential for danger is really high. This is what autoimmune disorders do. They attack your own cells because the cells that you've got, these B cells and T cells, don't mark off the right things. So we can't just release them out into the wild. We have to send them off someplace where they're going to be isolated and contained, but also able to learn, right? To mature, to become the kinds of cells that we need them to become. And that's what lymphoid tissue really is for us. Okay, a place where these cells can go, can learn in a safe environment, and eventually mature and be active parts of our immune system. There are also going to be places where we undergo what we call surveillance, right? Because all of your lymph passes through your lymphoid tissue, it's a great place to kind of keep an eye out for macrophages for pathogens and things like that, things that we need to be on the lookout for, cancer cells, all that kind of stuff. When we talk about building lymphoid tissue, it's mostly reticular connective tissue, right? This loose connective tissue, and it occurs in basically two kinds, right? We're going to have follicles, which are tightly packed bundles of lymphoid tissue, right? And in the follicle, we're going to find B cells here. And then we're also going to find, excuse me, we're also going to find less tightly packed, more sort of loosely arranged lymphoid tissue that we call diffuse lymphoid tissue. This diffuse lymphoid tissue is basically everywhere, okay? In all of the membranes of your body, surrounding every body organ, okay? Places where we have the most bits of this diffuse lymphoid tissue, there we go, are going to be in the mucous membranes. And if we think back to what a mucous membrane is, that makes a ton of sense, right? Because a mucous membrane is going to be lining a body cavity that's exposed to the outside world. That's how these pathogens get inside your body, right? They come in through your mucous membranes. That's why we're telling you not to touch your damn face right now, right? Because your face, your mouth, your nose, your eyes, your ears are basically lined with these mucous membranes, right? Which are great avenues for pathogens to enter your body. Things like the coronavirus that we're dealing with right now, COVID-19. So because these are such likely places for things to come in, this is also a place where we're going to have a lot of this lymphoid tissue so that we can at least try to get a head start on trying to deal with the pathogen. Once it's in there, it's already too late, you've got it. But this is a way for us to at least begin starting to try to mount a response to it once it's already in. Follicles, on the other hand, right, again, these are tightly packed lymphoid tissue. It's not diffuse at all. And they typically are going to be restricted to relatively small parts, right? So we'll talk about lymph nodes. We'll see them in we'll see them in a lot of our different lymphoid organs as we move forward. We'll see them in the back half of your small intestine in the form of Peyer's patches, okay, and also in your appendix. And the reason we have these here at the back half of your small intestine and in your appendix is that your colon, which is the immediate other side of your small intestine, has a lot of bacteria in it, like on purpose, right? These bacteria are how we produce things like vitamin K, right? We don't make it on our own, but we get it from these bacteria. You need to have a certain amount of bacteria in your colon in order to kind of be perfectly healthy. But we don't want them anywhere else, right? And so they have a sort of a potential for coming in kind of the back door, right? Leaving the colon and entering the small intestine. And so these Peyer's patches are there to kind of monitor that. And the same deal here with the appendix. Your appendix is not a useless organ. Is not a thing that we don't have any idea what the purpose is. The appendix, actually, we do know what it is there for. It's a place where these bacteria can grow, right? So that as we shed them through defecation, as we maybe have them die off due to antibiotics or anything else along those lines, we have a chance of recolonizing our colon. And so your appendix does that for us, but it's also there, of course, to try to monitor what's happening there and to produce some B cells to make antibodies against those things so that if they do get into your body, we can very quickly get rid of them. Now, lymph nodes, these are going to be kind of the principal lymphoid organ of the body, right? As it says there on the slide, obviously. They're all over the place, but they tend to be isolated to a couple of different spots, okay? So where your leg meets your torso, in the inguinal region here. So if you ever had a hernia check, this is what you're checking for there, an inguinal hernia. This is the region through which the testes descend and actually enter the scrotum if you're a male. A lot of blood vessels and things very close to the surface here. So of course these lymph nodes also come with that. Also in the armpit, the axillary region, and in the cervical region, so up underneath your chin here are places where lymph nodes will come, close to the surface. So if you go into the doctor and they're trying to diagnose what is wrong with you, they will probably feel at least your cervical lymph nodes, maybe your axillary lymph nodes, maybe your inguinal nodes, depending on the disorder, because these will swell with fluid as pathogens gather in there and we try to expose more B cells to them. So they will tend to swell and become exposed as a part of your natural immune response. The Black Plague, for example, the reason they called it the Black Plague is that you would develop what are called buboes, little owls. In your axillary nodes, they would swell and turn black, hence Black Plague. And of course, Black Plague is still around. If you go to Arizona or New Mexico, Black Plague is hanging out there, but we're better off now than we were in the 14th century. As far as what lymph nodes do, they filter the lymph, right? So all of your lymph passes through these lymph nodes. And so there's all kinds of trash that's in there, right? It doesn't have to be pathogens, right? It can be broken down bits of cell. It can be worn out stuff. It can be stuff we just need to just get rid of, right? That we don't want to dump back into the blood because it's trash. So that's a part of what we do with it. The other part of it is because we have all of these follicles within the lymph node itself. Again, these are, if you have a follicle, you have B cells. And so this is a place where lymphocytes, B lymphocytes, can be exposed to pathogens and begin to mount their defense or attack against those specific pathogens. As far as their structure here, and this is the last thing we'll cover in this video, as far as the structure here, they're mostly kidney bean shaped, right? They can be kind of roundish, varies a little bit, it's not terribly important. But the overall structure is going to be important because it's going to be very, very similar to all of our lymphatic organs. They're all going to be kind of built around the same basic idea. And that basic idea starts with an external fibrous capsule that has little walls that come inward from that fibrous capsule called trabeculae. And these trabeculae serve to divide the inside of the lymph node or the lymphatic organ, whatever it is, into little compartments, into little rooms. Okay, and then in the very center of this lymph node, we're going to have sort of an open area called the medulla. So the outer part with the trabeculae, with these nodes and the follicles, is the cortex. The inner, more open part is the medulla. Cortex is where these follicles are, right? So these are the B cells are, okay? There are also going to be some T cells in here, but they are not living here, right? They are moving through. It's like you play Monopoly, right? B cells are in jail, T cells are just visiting. Okay, they're not hanging out there, they're not living there, they're just passing through. And if we look at how this thing is arranged, okay, we've got, again, this outer fibrous capsule, these inner trabeculae that isolate out follicles. Okay, so we've got these structures here full of follicles, all of our B cells hanging out in here, and then our inner, more open area, okay, the medulla. Fluid, lymph, comes in through these afferent vessels, right? Filters in through each of these chambers, passes through each of these cortical regions, passes past all of these follicles, okay? Eventually filters in here to the medulla, and then out through these efferent, right? Obviously just efferent, but with an E, vessels that leave then the lymph node and continue on their way. I don't have to worry about that. This is just a actual picture. I like to show actual pictures when I can. It's pretty similar to the cartoon, right? With our fibrous capsule and our trabeculae and our little follicles and all that kind of good stuff. You can see here a little better now, kind of the more open medullary region versus the sort of more tightly packed cortical region. But that's really all that we're getting out of that. Now, the idea behind this, right, with all of these indoors and only a couple of outdoors, is that fluid backs up inside the lymph. The lymph node, excuse me. It's like if you've ever been to Disney World, right? Obviously Disney World's closed now, but if you've ever been to Disney World and you go on the Haunted Mansion ride, you go in first and there's all these doors. So everybody flows into this big open room, right? And they close those doors and you sit and you watch a video and this is the whole thing, right? But when it comes time to get on the ride, there's like one door, okay? And so while everybody got in the room really quickly and you felt like you were really making progress, it takes you a really long time to actually get on the damn ride, right? The same thing is true here with our lymph. It comes in very easily. There are all these doors, all these ways in, okay? But only a couple of ways out, right? And so it tends to hang out inside the lymph node for a long time. And the reason for that is the same reason we want to slow things down in the capillary beds, right? It takes time to do what we need to do. The longer this lymph stays in the lymph node, the more likely we are to, say, notice a pathogen and expose a B cell to it. The more likely we are to get rid of some cellular garbage that we need to destroy, whatever else, right? It needs to come through here and pool and sit and not just continuously flow. Right. So again, that comes down to just having very few exits and a lot of entrances. Okay, so when we come back in the next video, we'll talk about the spleen. From there, we'll finish off with the rest of the lymphatic organs, and in the next set of videos, we'll actually talk about the immune system.